When and then. All right. All right. Thank you all for coming. This is our first seminar for semester, right? First one this semester, and it is my pleasure to welcome Dr. Jason DeBoer. Just remember that maybe last time we met, Dr. DeBoer and I said something like, Dr. DeBoer has a lot of experience with, being out in the field and doing a lot of research. And he said, "Are you calling me old?" I was not. But I just talked with him about his background, and he has done a lot more than I have. So let me go over some of these things. Originally from Kentwood, Michigan, he got his undergraduate degree from Grand Valley State University in Michigan, then worked as a residential and commercial tree kind of work for 4 years, went back for his master's at, again, Grand Valley State University, then got another master's from University of Nebraska, Lincoln. He started his current position at IRBS in 2013, and he has been there since. Starting 2016 and ending in 2020, he got his PhD from University of New England in New South Wales, Australia. And all this time, he has been working at IRBS, I would assume, at different positions, right? So it is, again, my pleasure to welcome Dr. DeBoer. Take it away.
Thank you, Bhaskar. And thank you all for coming today. Make sure I get everything clicked on correctly here. There we go. Before we get started learning about resilience in Anthropocene rivers, you need to meet your river guide for today, and that's me. As Bhaskar mentioned, I grew up in Kentwood, Michigan. I lived there until I was about 30 years old, right where the little orange star in the mitten is. Growing up, I was a lot, this is not me. We didn't have color photography when I was a kid. I was a lot like the little boy in this picture. I loved spending time outdoors, splashing in the creek that ran near my house, and generally getting as wet and muddy as I could most of the time, which honestly describes the 45-year-old me pretty accurately too. I earned a bachelor's degree in natural resource management from Grand Valley State University in 2001, and a master's degree in biology from Grand Valley in 2006. For my master's work, I worked in partnership with the Little River Band of Ottawa Indians near Manistee, Michigan, focused on stream habitat restoration in Sickle Creek and Bear Creek, with emphasis on a little fish called a mottled sculpin in Sickle Creek. So the Little River Band got a grant to remove outdated and ineffective road culverts on the reservation and replace them with more environmentally friendly open bottom bridges. We measured changes to the stream bed and the response of aquatic insects and fishes to those changes. We also tracked the movement of mottled sculpin, which is that cute little fish in the bottom right corner, in those newly restored stream reaches. After finishing at Grand Valley, my wife Sue and I moved to Lincoln, Nebraska, where I started a graduate program at the University of Nebraska-Lincoln. That project focused on environmental factors affecting recruitment and growth of walleye and white bass in Southwest Nebraska irrigation reservoirs. It was beautiful country, but it was totally different than where I'd grown up in Michigan. I worked with larval walleye only a few millimeters long and found out that they still have teeth at that size. The fish in the top middle there is a ½ an inch long, 12 millimeters, and still has teeth. We also worked with great big trophy walleye, as you can see in the top right-hand corner there. And part of my research there involved figuring out hatch date from the larval fish that we captured using daily rings on otoliths, which are bones in a fish's head, like you see in the bottom left-hand corner there. Each one of those rings is 1 day of growth for that particular fish. We also wanted to figure out how old some of the larger fish were, and we used one of the dorsal spines off the fish's back, as you can see in the bottom right-hand corner there. So rather than daily rings, those are annual rings, kind of like you'd see on a tree if you cut a tree down and look at the rings on a tree. After finishing in Nebraska in 2013, Sue and I and our new dog, Abby, moved about halfway back home to Michigan and landed in west central Illinois, where I got a job at the Illinois River Biological Station, or IRBS for short, one of 7 field stations operated by the Illinois Natural History Survey, part of the U of I. Most of my work involves adding to long-term fish population monitoring data sets using this big electrofishing boat. Yes, in a sense, I get paid to put electricity in the water. I worked my first 6 years on the Illinois River from Chicago to St. Louis, the last 5 years on the Mississippi River from the Quad Cities down to Quincy or Hannibal. As Bhaskar mentioned, while I was at IRBS, I had an opportunity to get a PhD in interdisciplinary river science from the University of New South Wales University of New England in New South Wales, Australia. My PhD project focused on large-scale changes that we as people have made to the Illinois River and its watershed over the last 200 years, and how those changes have affected its ecosystem. Most of the changes that we as people made to the Illinois River were a consequence of the Chicago Sanitary and Ship Canal, which was built, as you can see it from these pictures, in the late 1800s. That's 1895, not 1995 in the top left there. These changes included a system of 8 locks and dams and also a network of levees which protect valuable farmland, but also separate the river from the floodplain. I'll get into a lot more detail about that research in just a little bit. And that brings me to my seminar title for today. The presentation I'm sharing with you all is the culmination of my PhD research at the University of New England. Lest you think I completed all of this on my own, let me take a minute to acknowledge those who supported this process. My multi-continental supervisory committee were obviously influential in guiding me throughout, and our station director, Jim, supported me in finishing my degree when he joined our station halfway through my program. All right, diving right in now. Large rivers are critical social ecological systems, providing drinking water, transportation avenues, and productive inland fisheries. Those ecosystem services alone are worth more than $4,200 per hectare per year, which makes large rivers one of the most productive ecosystems on the planet and the most affected by humans. Despite how much we value large rivers, we often don't treat them very well. There are few ecosystems on Earth that have experienced more anthropogenically driven change than large rivers. Multiple anthropogenic stressors have decreased the productivity and resilience of large rivers for over a century, negatively affecting these valuable ecosystems. More than 65% of the world's large river ecosystems have been fragmented by dam construction and downstream flow regulation, while dredging, riverbank protection, and leveeing have resulted in system-wide simplification of once diverse and dynamic riverine habitat. So what we're going to do today is use resilience thinking like a big lens through which we'll look at a large river ecosystem. We'll use this lens to examine whether a large river ecosystem has shifted or flipped from a natural or pristine regime to a new regime. But first, I need to spend a little bit of time explaining how we believe the concept of resilience thinking works. Resilience thinking is a novel approach to studying systems and disturbance in systems, and provides a good theoretical foundation for understanding how ecosystems recover from or change in response to local disturbances like floods and system-wide disturbances like climate change. The concept of ecological resilience assumes the presence of multiple regimes and the tolerance of the system to disturbance that triggers shift between regimes. In 1973, Dr. Buzz Halling defined ecological resilience as a system's ability to absorb change and disturbance and still maintain the same relationships between populations or state variables without crossing a threshold, as you can see in the middle of the figure there, and moving into a different regime. In 1996, Dr. Halling further refined his definition of ecological resilience as the amount of disturbance that a system can absorb without changing its structure and function. Thus, for the purposes of my research and my presentation today, resilience is the capacity of a system to absorb stressors and remain in the same regime. That is, to retain the same structure and function. With that as context for my presentation today, here's the plan for our journey down the Illinois River. First, I'm going to introduce all of you to my beloved Illinois River. Next, I'm going to demonstrate how ecosystem structure and function in the Illinois River have changed over time. Then I'm going to describe the character of that new regime. And finally, I'm going to end with discussing some philosophical and practical implications from this research. The first leg on our journey is to introduce the Illinois River, a 500-kilometer long system that forms at the confluence of Des Plaines and Kankakee rivers. It flows from near Chicago, Illinois to St. Louis, Missouri, and provides connection between the Great Lakes watershed and Mississippi River watershed. The river is home to nearly 200 species of fish, around a dozen of which are non-native. The river is most often separated at the Great Bend into general geomorphic reaches. The upper river is higher gradient with coarser substrates and a narrower floodplain with fewer natural backwater lakes and in-channel islands, whereas the lower river is a lower gradient with finer substrates and a wider floodplain, which historically had more backwater lakes and in-channel islands. A fun fact for you, the lower Illinois River channel from the Great Bend downstream is actually a historic abandoned channel of the Mississippi River. The Illinois River is an excellent choice to study because of a long-standing legacy of major abiotic and biotic anthropogenic stressors. For example, common carp was introduced in Illinois in the year 1890. This non-native barbelled menace forages in benthos, increasing turbidity, damaging habitat, and competing with native fishes. Arguably the most important stressor, the Chicago Sanitary and Ship Canal, opened in 1900. It connected the Great Lakes watershed to the Mississippi River watershed by reversing the flow of the Chicago River and essentially flushed Chicago's sewer down the Illinois River. This created a huge influx of municipal and industrial waste that resulted in horrible water quality for over half a century. In fact, the water pollution was so bad that in 1920, the river was anoxic and biologically dead for 200 kilometers downstream of Chicago. Thanks to several INHS scientists, we have dissolved oxygen data from the late 1800s to present for the Illinois River and can show just how bad things got, with DO levels below 2 milligrams per liter for near half a century. For reference, anything below 5 milligrams per liter, which is shown by this white dashed line here, causes stress or death in aquatic organisms. We were way below that for more than 50 years. The Des Plaines and upper Illinois River near Chicago had nearly uninhabitable dissolved oxygen levels for more than 50 years. Getting back to our timeline now, a series of 8 locks and dams was built on the river during the 1930s. Now that facilitates barge traffic, but it also unnaturally stabilized the river's hydrology, the seasonal fluctuation of floods and droughts, and also fragmented longitudinal river connectivity. But it's not all bad news. Now we have some good news. The Federal Water Pollution Control Act of 1948 was the first major US law to address water pollution. Growing public awareness and concern for controlling water pollution led to the act being amended and expanded 6 times between 1948 and 1987, with the large-scale 1972 amendment known as the Clean Water Act. I heard it. The Water Pollution Control Act, Clean Water Act, and other amendments were system-wide attempts to improve water quality around the country. Although the benefits were dramatic and most types of water pollution declined, today more than half of US streams and river miles continue to violate water quality standards. Nonetheless, water quality in the Illinois River improved a lot in the 70s, 80s, and 90s. But more bad news. 25 years after the Clean Water Act, just as conditions were improving, invasive carp species began taking over the Illinois River, imported from Asia to clean algae from southern US fish farms. Silver carp, as you see in the picture here, and bighead carp are large-bodied, fast-growing planktivores, which are incredibly invasive and highly disruptive to the base of the food web in most Midwestern US rivers. With that as an introduction to the Illinois River, let's start the second leg of our journey, which focuses on ecosystem structure. My aim in this leg of the journey is to examine the response of fish biodiversity, which is one measure of ecosystem structure. Measure that response to anthropogenic stressors at a long temporal scale and a broad spatial scale. We're going to use long-term data to evaluate the response of our river system. How does the river respond? Does it respond positively, negatively, and at what rate? Moreover, does that response vary by the location in the river network? To investigate these questions, we used the electrofishing data from the long-term electrofishing program, the program that I've worked on at IRBS for the last 12 years. It's a long-term system-wide fisheries data set that runs from 1957 to 2015. That's nearly 60 years of data from an entire river, making LTEF one of the longest running aquatic monitoring programs in the country. Surveys were conducted annually at 27 different sites, shown by the yellow dots on the map there, along the entire length of the Illinois River. To analyze that, we used Sizer, which is a powerful breakpoint analysis tool to look for statistical changes and trends over time in the slope of fish diversity data, which we believed would influence or reflect the influence of those anthropogenic stressors. We want to know, can the system even respond, or is it too degraded? If it can respond, when did it respond? What is the direction, positive or negative, of the response? And did that response change from upriver to downriver? Did it vary spatially? We performed separate analyses for the whole river and then those two river zones, the upper river and the lower river, which vary geomorphically. We did a lot more and different analyses for this research, but for the sake of time today and not boring you with statistics lecture, I'm only going to show you the basics. This will be the standard format for all of the plots that I'll show you in the next few slides. We have Shannon Diversity Index on the y-axis and year on the x-axis. This plot is for the entire river, so both of the river zones, and then all fishes combined. We didn't break it down into different taxonomic groups. And what we see from 1957 to 2015 is that overall fish diversity did increase through time. We have two different breakpoints in slope depicted by these orange vertical lines in 1970 and 1985. Now, if we separate spatially, the first graph was the entire river. This page shows the upper river and the lower river separately. Again, these are different geomorphic zones. We can see that the diversity of all fishes in both the upper river and the lower river also increased through time. So the response through time was similar in space. Those breakpoints are also the same as they were for the whole river, 1970 and 1985. We found it interesting that we have synchrony in the timing of breaks for the entire fish community at two different spatial scales. To summarize these really brief results, despite a legacy of negative anthropogenic stressors, the system was able to respond. We observed an overall increase in diversity with significant breaks in slope for all of those line segments. The take-home message from this leg of the journey is that fish diversity as a measure of ecosystem structure has changed over time. Because of the persistent and likely permanent presence of introduced and invasive species, the response of native fishes to the legacy of system-wide anthropogenic stressors like locks and dams and levees, diversity cannot go back to what it was before, so it has permanently changed. So now that we know that ecosystem structure as measured by fish diversity has not only changed, but can't go back to what it was before, looking through this lens of resilience, these differences in ecosystem structure provide our first line of evidence that a shift in the Illinois River from a natural or pristine regime to some kind of new regime. The third leg of our journey down the river today focuses on ecosystem function in the Illinois River. This slide shows the timeline I showed you earlier, separated into four distinct phases of system change in the Illinois River: settlement, degradation, recovery, and biological invasion. Each phase reflects a different set of system-wide changes from the late 1800s to the present. The need for these separate phases will become clear in just a little bit. The settlement phase is our reference condition. That was a period of minimal impairment which ended in 1900. The degradation phase began in 1900 with the opening of the Chicago Sanitary and Ship Canal and locks and dams and levees built until the 1930s and lasted until 1948. The recovery phase began in 1948 when the Federal Water Pollution Control Act was passed, with the Clean Water Act passed in 1972, and that phase lasted until 1996. The biological invasion phase began in 1996 when zebra mussels and big-headed carps and other species invaded the river, and that phase continues until today. So if we want to investigate differences in ecosystem function due to stressors going back 150 years or more, how in the world do we get samples? Well, biological collections housed at universities and in museums are often able to be used for this purpose and can provide us with a chronology of stable isotope samples that can be used to investigate differences in food webs, which is one way that we can study ecosystem function. In our case, The spatial and temporal range for which samples were actually available from the Illinois River only allowed for comparisons of the upper and lower river zones that differ in geomorphology and those four phases that I just described, which captured periods of different local and system-wide stressors. So we didn't have a continuous data record, but we had enough samples in enough of those phases that we were able to compare categorically among those four different phases. Our aim on this leg of the journey is to determine if there is a sustained difference in ecosystem function as opposed to ecosystem structure among those timeline phases as measured by food webs in the Illinois River ecosystem. Again, we're using the settlement phase as our reference or natural condition, given the minimal impairment to the river during that phase. We're going to use long-term and large-scale stable isotope data to evaluate the response of ecosystem function along the Illinois River. How does it respond? Again, does that response vary by location within the river network? And one more layer, does that response vary among different functional feeding guilds? We collected archival and modern mussel, fish, and snail specimens. We used the mussels and snails as surrogates for pelagic and benthic carbon sources. And we collected fish from four different functional feeding guilds: piscivore, meaning they eat other fish; invertivore, which makes Manisa shudder because she studies macroinverts; omnivore, which means they'll eat anything; and planktivore, which means they eat zooplankton or phytoplankton. We studied four different metrics to show differences in ecosystem function in the Illinois River over that 150-year period. We looked at bulk stable isotope ratios, community niche space, basal resource contribution to higher levels in the food web. So are you getting your food from a planktonic or demersal source? And then mean trophic position of fish. Today, I'm only going to show you the first two, I think maybe three of these for the sake of time. I think I weaved food web models in there. So just a one-slide intro to stable isotope biplots for those of you who are unfamiliar. Stable isotopes of carbon and nitrogen can be used not only to assess the relative contributions of pelagic and benthic basal resources, but also to provide information on trophic position. So carbon is displayed on the x-axis and is used to describe basal energy source, which is where the critters get their food from. We have pelagic on this side and benthic on this side. So if it's plankton derived, it's growing over here. If you've got leaves and things falling in the stream and that's where they get their carbon from, it shows up over here. Nitrogen is shown along the y-axis, and that's used to describe trophic position, which basically means how high up on the food chain you are. So we have phytoplankton down here, and then we'd have piscivorous fish or eagles or osprey or people at the top of the food chain up here. Before I show you everything, we'll start with just isotope biplots for carbon and nitrogen for the two basal sources in the upper river. Again, carbon is on the X, nitrogen is on the Y, and the different symbols depict the different phases. That's a little small, but I coordinated the colors for you to kind of counteract that. The orange circle is for settlement. That's our reference condition. The downward triangle is degradation. Things are getting worse, so they're going down. The upward triangle is recovery. Things are getting better. They're going up. And then biological invasion is the diamond phase. So what I want you to notice on this slide and on the next slide is that the orange circles from the settlement phase, this orange circle here and this orange circle here, see that most of the other shapes are distinct from them. There's no overlap in those confidence intervals in either direction. So the orange shape, separate from everything else. That's what I want you to focus on, because I'm going to overwhelm this slide with a bunch of more graphs. This slide shows all of those trophic groups separately. So we have mussels and snails on the top. We've got the upper river on the left, the lower river on the right, and then those four taxonomic groups for the fish below that. But what you can see is that only one of the guilds has returned to the values found during the settlement phase, which is again what we're using as a reference condition. That was upper river planktivores, which is here. You can see that orange dot has been occluded by the diamond from the biological degradation phase. In every other graph, except invertivores, that was the same during recovery. The orange dot is distinct from all of the other shapes, meaning things are different now than they were in that settlement phase before 1900. This is our first line of evidence that ecosystem function is different among these timeline phases. This slide combines the four gills and two basal sources from each phase. So essentially what I've done is take all 6 of those plots and crunch it into one plot. Again, we have carbon on the x-axis and nitrogen on the y, and the different symbols again that depict the different phases. So this is called a convex hull, and what this depicts is differences in shape, area, and most importantly, position between each of the four phases. As you can see, the orange shape is on the bottom of the graph, and there's this vertical progression, an increase along the y-axis for all of the other phases. The lower river on the right shows a similar sustained movement up and away from the settlement phase, as does the upper river on the left. This is a second line of evidence that ecosystem function is different among timeline phases, and we did not return to things in the settlement phase. So 4 separate lines of evidence, the 2 that I showed you, the 2 that I didn't, indicate substantial, significant, and sustained change in food webs among the 4 timeline phases. System-wide attempts at restoration, like the Clean Water Act, were effective at improving water quality, but were unable to return ecosystem function to what it was during the settlement phase. Moreover, additional stressors like invasive species continue to cause new differences. Several invasive species, primarily silver carp and bighead carp, have caused differences that result in furthering the development of a novel ecosystem, particularly in the lower river. The multiple lines of evidence that I shared show important differences in ecosystem function in the Illinois River. Moreover, the river did not return to pre-stressor conditions following major attempts at restoration. Looking through our resilience lens again, these sustained differences in ecosystem function among phases along the Illinois River suggest a shift from a natural or pristine regime to a new regime. Recall the definition of ecological resilience I gave you earlier in my presentation today, the ability of an ecosystem to absorb stressors and retain the same structure and function. So we first learned today that ecosystem structure in the Illinois River is different because fish biodiversity has changed over time. And we just learned that ecosystem function is also different according to multiple food web metrics. Taken together, these sustained differences in ecosystem structure and function indicate that when looked at through a resilience lens, the Illinois River has flipped to a new regime, which will have implications for its future management and attempts at rehabilitation. More importantly, the Illinois River can't go back to the previous regime because of permanent system-wide changes to its physical environment, including levees, locks and dams, and channelization. Viewed through that resilience lens, anthropogenic stressors have flipped large river ecosystems all around the world into a new regime where they exhibit different structure and function. At a global scale, humans have affected physical landscapes, atmospheric processes, and climate. Humans have also affected animal biodiversity and even genetic diversity, especially of fish and insects. From a theoretical standpoint, these regional and global scale human-induced environmental modifications mean that the Earth has undergone sufficient change to imprint a new global geographic signature, triggering a new epoch. According to numerous scientific societies, we are in the Anthropocene. So that's a fun buzzword for the geologic epoch that we're in now, the Anthropocene. But what does it actually really mean in a context that's meaningful to us when we study and then have to manage large rivers? Put another way, how are Anthropocene rivers different from natural large river systems? To answer that question, we're going to explore the Anthropocene Illinois River's character, not only that of the physical environment, but also the interactions between its physical environment and 2 measures of ecosystem function, food webs and fish growth. So in large rivers, the physical environment provides this template that kind of shapes an organism's life history. This means that the physical character of river networks determines patterns in the abundance, arrangement, and type of biological assemblages within riverine landscapes, and also how these assemblages function and interact. Conceptual models that attempt to illustrate these patterns and their interactions include classic concepts in river science like the river continuum concept and the flood pulse concept. The riverine ecosystem synthesis is a more recent approach that proposes the river network consists of functional process zones, or FPZs, which are physically distinct, large-scale geomorphic patches where ecosystem structure and function vary between FPZs. Previous studies in other rivers have shown that FPZs have unique riverbed habitat, fish community, benthic macroinvertebrate community, and food webs, which, just as we learned, is one measure of ecosystem function. I'll mention it again because it's important going forward. In relatively unaltered rivers, previous studies have shown measures of ecosystem function are different among FPZs. So I've shown you this map of the Illinois River a couple of times today. And for this part of the project, my co-author and advisor Martin did a bunch of fancy GIS modeling to not only identify FPZs for the Illinois River for periods before 1900 and after 1950, but also to calculate the heterogeneity of those FPZs. So heterogeneity for our purposes has three different components: composition, configuration, and character. For our purposes, composition is the number of FPZs in a river. Configuration is their spatial arrangement along the river. And character is the physical heterogeneity of each FPZ. Features like the shape of the river channel and the riverbed, and also substrate complexity. So here's our timeline once again, this time with the pre-1900 period identified as the natural regime, and the post-1950 period after the opening of the canal and most of the construction of the locks and dams and levees identified as the Anthropocene regime. Our aim for this leg of the journey was to answer three different questions. How does heterogeneity, once again, composition, configuration, and character of FPZs differ between the pre-1900 and the post-1950 regimes? What's the interaction between FPZs and ecosystem function? And third, what is the heterogeneity of ecosystem function in the Anthropocene Illinois River? Here I've overlaid the 6 different FPZs that Martin identified along the length of the Illinois River. And as you can see, they don't just correspond to the locks and dams, which are those thicker black lines bisecting the river. Importantly, the composition and configuration of FPZs along the Illinois River did not change between pre-1900 and post-1950 periods. We have the same number of FPZs, they're in the same places, and what did change was the physical heterogeneity within each FPZ. That has decreased, as you can see by the graph, particularly in FPZs 1, 2, and 3. As a result, the Anthropocene Illinois River channel exhibits a system-wide decrease in physical heterogeneity after the year 1950, meaning a simplification of the river's landscape relative to what it was naturally before 1900. So what does a simplification of physical heterogeneity look like? Not on a bar graph, but in an actual large river landscape. Simplification includes factors like straightening river channel plan form via channelization. It includes decreasing riverbed complexity and cross-sectional morphology via sedimentation. So things have been straightened out and filled in. We've lost coarse substrate, we've lost complex river channels. So now that we knew we had the same 6 FPZs in the same places as we did pre-1900, we had to determine if there was an interaction between modern FPZs and ecosystem function, and then determine the heterogeneity of ecosystem function within the Anthropocene Illinois River. We used the same suite of food web metrics as we did for the previous leg of our journey, comparing ecosystem function among time phases, but this time we collected modern specimens for each of the 6 FPZs. First and most importantly, a strong positive relationship was apparent between FPZ physical heterogeneity on the x-axis and food chain length on the y-axis. Previous research has shown that food chain length is one attribute of ecosystem function that is strongly affected by anthropogenic modifications to river hydrology and geomorphology. Importantly, this strong association suggests that simplification of the physical environment we found could negatively affect ecosystem function. These are isotope biplots, and rather than orange and white, the 6 different colors here represent the 6 different FPZs that we found along the length of the Illinois River. The important pattern to notice here is that the basal sources, mussels and snails, which are in the 2 left-hand plots, all show spatial separation, whereas all the other high-level consumers in the other 4 plots exhibit a fairly high degree of clustering of isotope ratios. So everything on the left, the 2 graphs on the left, mussels and snails, there's spatial distinction. The 4 graphs on the right, there's a much higher degree of clustering. The dots are a lot closer together. This clustering of higher level consumers is our first indication that simplification of the physical environment in the Illinois River could be causing homogenization or conglomeration of ecosystem function. Next, we looked at the community niche space of the 6 FPZs, and we saw that significant overlap exists among those FPZs. All of these different colored polygons have a lot of overlap. In natural or less disturbed rivers, previous studies have shown that polygons like this are separated among FPZs. But disturbances like invasive species, environmental degradation, and urbanization can cause them to overlap. This high degree of overlap is our second indication that simplification of the physical environment of the Illinois River could be causing homogenization of ecosystem function. This slide showed food web models. I thought I had food web models in the first part. I have food web models over here. This shows food web models for each FPZ denoted by the number on each plot. And again, I know this slide is busy, but two things to focus on here. First, that trophic position of all of the consumer groups really only shows two distinct groupings. We have FPZs 1 through 3 on the top with an average around 3. So if you find 3 on each graph, most of these are about at that level. There's not much variation between FPZs 1, 2, and 3 in those trophic groups. FPZs 4 through 6, on the other hand, have an average around 5. As you can see, that number 5 really runs through right about here. So we have distinction between FPZs 1, 2, and 3 and FPZs 4, 5, and 6. We really only have 2 groups, whereas we should have 6. The second thing I want you to focus on is that benthic sources are the dominant basal resource for higher level consumers in the food webs of all of the FPZs on the river. So each graph has a B and a P. We have benthic and pelagic. And you can see the B is a bigger number than the P in every single one of those graphs. In the FPZs from the highly turbid lower river, we'd expect to see more pelagic contributions as turbidity inhibits benthic production and allows pelagic or planktonic production. Although invasive silver carp and bighead carp may be causing a benthification of the lower river's food web. So in a natural river, we would expect the P in the lower river, FPZs 3 through 6, to be larger given the turbidity there. Both of these points of emphasis are further indications that systemic simplification of the physical template in the river could be causing homogenization of ecosystem function. So to summarize this leg of our journey, we documented a loss of heterogeneity of the physical environment between the natural and Anthropocene river networks, or a system-wide simplification of the river's physical environment. We also documented a strong positive association between the physical heterogeneity of each FPZ and the food chain length in that FPZ, which again is a measure of ecosystem function. Based on that strong positive association, we demonstrated a homogenization of ecosystem function using multiple lines of evidence from food webs, which highlights a link between simplification and homogenization, which I'll come back to in just a bit. So through that lens of resilience, we've learned about why the Illinois River is in a new regime as an Anthropocene river, and we've learned about the character of the river's physical environment and ecosystem function in that new regime, which means there's just one more point I'm going to emphasize today, that being the management implications and applications of this research before we reach the end of our journey today. So what does a link between simplification of the physical environment and homogenization of ecosystem function mean, at least in a philosophical context? Simplification of large river landscapes at broad spatial scales reduces the number of niches and consequently affects species diversity, which is ecosystem structure, and also ecosystem processes, which is ecosystem function, which then decreases redundancy and resilience in the system. Essentially, this means that Anthropocene rivers have a reduced adaptive capacity. They've lost the ability to adapt to additional natural disturbances and anthropogenic stressors. From a resilience thinking perspective, a loss of adaptive capacity infers that regime shifts in Anthropocene rivers may become more common in the future. Showing that a loss of adaptive capacity in the system can result from simplification of the physical environment at the whole river scale is really interesting. Using resilience thinking's beloved ball and cup model, where a system is represented by a cup and the system state is a ball rolling around in that cup, we have shown that simplification of the whole river or the whole cup has reduced the amount of disturbance needed to cause a regime shift or to push that orange ball from one state to the next. Note that the size of the arrow is decreased. Where's my pointer? There we go. Big arrow here, little arrow here. We have deep cups in a complex system. We have shallow cups in a simplified system. So we've shown that the theoretical principle of simplifying the surface in the ball and cup model actually translates to simplifying the physical template of the river ecosystem. That's one of the biggest philosophical contributions that our research makes. However, a loss of adaptive capacity infers that regime shifts in Anthropocene rivers may become more common in the future, especially considering the threats of climate change and invasive species. Although that seems pretty bleak, we have the ability to prevent this. There are levers that managers can pull to increase the adaptive capacity of Anthropocene rivers so they're more resilient and don't undergo further regime shift. We think that there are two main levers that managers can pull, and both should be used together when possible. The first lever, flow management, is highly touted and often emphasized at the expense of the second, especially in rivers with hydropower regulation. We believe more focus needs to be on that second lever, heterogeneity, especially at the whole river scale. Many studies suggest restoring rivers could be facilitated by allowing for a quote-unquote more natural flow regime. That was a concept popularized by Leroy Poff and others in 1997, and it's now often conflated with environmental flows, which is something you hear about in Western US rivers. Restoring a natural flow regime should theoretically benefit hydrologic and geomorphic processes and also benefit biota. Much of this idea, like I said, was popularized for mountain rivers, mountain rivers in the Western US, but there are still options for those of us in the Midwest. Environmental pool management was implemented in the late 90s by the Army Corps of Engineers on pools 24, 25, and 26 of the Mississippi River near St. Louis. And annually, it creates thousands of acres of critical habitat for fish and wildlife in these pools. By drawing down pool levels just a couple of feet for at least 30 days in the summer, which promotes aquatic plant growth in shallow areas, while still maintaining a safe and dependable navigation channel, which is what the Corps of Engineers is tasked by Congress to do. The Corps uses an adaptive management framework to lower water levels between May and August, with that May to June period being the most desirable for vegetation growth and seed production. So if they can get below this full pool, a foot below that for 64 days or 2 feet below that for 50 days, we get this vegetation production in side channels while still maintaining a navigation channel in the river. So flow management is a good tool, but we believe efforts to manage and rehabilitate Anthropocene rivers must also pull a second lever and include attempts to increase physical heterogeneity at multiple scales in rivers. This can include reconnecting or improving off-channel or tributary habitats lost or degraded to anthropogenic stressors, and also improving in-channel habitat that's been similarly degraded. So increasing off-channel physical heterogeneity can be achieved by reconnecting historic channel meanders, as you can see in the left-hand image here. Similarly, as seen in the right-hand image, river tributaries are often relatively less altered than the large rivers that they flow into. Thus, their physical heterogeneity may be more easily restored than that of highly simplified large rivers. Increasing off-channel physical heterogeneity can be achieved by rehabilitating disconnected backwater lakes with managed water connections, which may return some of the functions and benefits of backwater lakes while still keeping invasive species out and better managing sedimentation. The Nature Conservancy's Emiquon Preserve is a 2,700 hectare complex near Havana, Illinois, right across the river from our office, that restored 3 disconnected backwater lakes that were levied off and in agricultural production for almost a century. Using a managed connection to the river through this structure right here, TNC produces fish and aquatic vegetation that can be released to the river, serving as a source for native organisms that can otherwise struggle to reproduce and recruit in the highly simplified main river channel. Rehabilitation can also include increasing heterogeneity of in-channel habitats. For example, island construction and side channel dredging can increase physical heterogeneity in rivers that have been simplified by anthropogenic changes. This temporal sequence here from 1939 to 2012 is from the lowermost 8 miles of Pool 8 of the Mississippi River near La Crosse, Wisconsin. This shows island erosion as a consequence of dam construction and barge navigation, and then island restoration for a habitat rehabilitation and enhancement project. Lock and Dam 8 began operation in 1937, so before this first image. Most of the islands, as you can see, were eroded away. There's no green in this image. Most of that was gone by 1989. And then island rehabilitation and construction was completed in 3 phases in 1993, 1999, and 2012, which is where we are today. Restoring the heterogeneity of those in-channel habitats is shown to increase flow diversity, as you can see from the same project here on Pool 8. You can note the increase in red, which are low flow areas in the right-hand image, showing the difference in flow diversity just between phases 2 and 3. Restoring heterogeneity of in-channel habitats also reduces wind fetch and thus large waves that can erode shorelines, uproot vegetation, and resuspend sediments. Reduced wind fetch is shown here in the increase in cooler blue and green colors and the decrease of the warmer pink and red colors, again, just between phases 2 and 3. Both increased flow diversity and reduced wind fetch benefit numerous river ecosystem components, including water quality, aquatic vegetation, macroinvertebrates, and fish. All right, that was a lot of information, but we have safely arrived at the bottom of the river today. To recap our journey, we've relearned that ecosystem structure and function in the Illinois River have changed over time in response to a legacy of anthropogenic stressors. Looking at these changes through a resilience lens means that the Illinois River is in a new regime, an Anthropocene regime, and can be called an Anthropocene river. We also learned that limited ecosystem heterogeneity is characteristic of the Anthropocene Illinois River, as evidenced by homogenization of food webs. This homogenization is associated with system-wide simplification of the river's physical environment. Simplification of the physical environment and homogenization of ecosystem function have implications for Anthropocene river resilience, as these rivers are highly susceptible to future stressors like global climate change and invasive species, which could cause them to flip into a new regime with even further reduced productivity because of lost ecosystem services. My take-home message today is that managers have multiple levers they can and must pull to restore physical heterogeneity at multiple scales, to reconnect off-channel habitats and improve in-channel habitats, and also incorporate flow regime management. All of these can work together to increase heterogeneity in ecosystem function, thereby increasing resilience to looming stressors like climate change, reducing the risk of further regime shift in Anthropocene rivers. To conclude, I think further studies are needed to better understand the role of ecosystem heterogeneity in absorbing natural and anthropogenic stressors in large rivers worldwide. Otherwise, there remains an undeniable risk of further productivity and biodiversity losses in these globally valuable ecosystems. So those last few slides that I showed you of Pool 8 in La Crosse, Wisconsin, where all the islands had been eroded and then came back, this is what the finished product looks like from an airplane. These are all those restored islands that were constructed in the bottom end of that pool. I thank you for watching my presentation, and I think I've got about 12 minutes left for questions.
You said early on is diversity can't go back to the kind of diversity it was before. And, you know, there's evident reasons for that, and you mentioned them as you went on. But in a bigger picture, how do you make a determination that, is there some criterion for saying that no matter what happens, what management we do, it can't go back to what it was before? Is it because there's non-native species now, or because those managements are unlikely to be done?
So the presence of the non-native species definitely hampers going back to that. It's so difficult to get rid of things like the invasive carps, but the permanent physical changes to the Illinois River, things like locks and dams and levees, we can't just take those out. The human infrastructure has been built up and around and is so dependent on those now that practically speaking, we can't get rid of them. And the system has adapted and evolved to be what it is in response to that, you know, species imprinting on that physical template. So.
This is about that same sort of thing. So I understood you used isotopes to see if food web's community function had returned. I saw that you had dots that data for pre-disturbance and then post-disturbance, and they didn't overlap. But for the species richness, so the Shannon,
Mhm.
the data you showed were like 1950 on. And I sort of was zoned out when you said we can't go back, which is what Dr. Hinkle was just asking about. But do you have data? Like, what's the Shannon index for 1890? So you said we can go back, but I didn't see the picture. There was a picture that says we're not back where we were.
No, I do not. We don't have that data. Species specific sampling, especially community level species specific sampling, was not nearly as prevalent in those periods as it was starting in the 50s. There may have been some surveys. We have records of some surveys, but Dr. Forbes collected 7 catfish and 3 largemouth bass. It doesn't tell us how long or where or anything like that. So we have records of some of the species that were present, but they're not translatable to the rigor that we have nowadays. So you can't compare richness and abundance and evenness, which are those measures that make up species diversity. So we can look at some of the presence absence, what species were there versus what species are not, and see that we have new things and we have lost things. So good question. Manisa,
Great presentation.
thank you.
About your increase in fish diversity, I've seen the graph is going up. So are you seeing mostly native species or non-native like transient species or invasive species? What's making it, high every year?
It's a little bit of both. We're seeing additional native species that were either absent in the 50s and are present now that were likely present before. So they're making a return, likely from further up in the watershed, whether that's coming down from tributaries or coming down from farther up. We're also seeing new and interesting invasive species pop up, things like weather loach, round gobies, some of those other Great Lakes invasive that are coming down. Ostensibly, we'll get fish from the Mississippi coming up the other direction. So the Illinois is kind of at that nexus of the Great Lakes ecosystem and the broader Mississippi River ecosystem. So we're getting a little bit of both. Yeah.
Just to follow up on that, so looks like there are more species coming in, or is it also because of your gears? The things have changed in this 50 years.
Yep. So the same gear was used for that entire fishery survey. Obviously, the specific boat changed, but the gear is the same between 1957 and 2015, which is what makes that data set so impressive. Things didn't change through space or time. It was sampled the same way, which makes these comparisons so powerful and so valid. Good question. You're welcome.
I was just gonna comment, that's super cool, but so you now restricted to using the same stuff they were using in 1950 in perpetuity forever?
Presentation I just gave last week talks about the transition between ending that particular program and moving on to a more fish-friendly and more widely accepted, widely used fish sampling technique. So we went from AC electrofishing, which is the electricity that's in your house or in the walls here, to a pulsed DC electrofishing. So what's more in your car's battery. The wave form is different. It's less injurious to fish. And it samples a slightly different component of the fish community, which is why that graph ended in 2015. We saved about half of the sites that were still sampleable. I talked about sedimentation quite a bit in this presentation. Half of the sites that we used to sample in the 50s, they're not even there anymore. They're either sedimented in, so bays have filled in or side channels have filled in, or the islands have eroded away. So there is no more side channel. So we saved about half those sites that we could use. We folded them into our new protocol. So we still sample those same sites, but do not use that same gear anymore. It was just too difficult to maintain. Dr. Jenkins.
Why did you use Shannon Weiner instead of a different person?
What I didn't show was that the abundance, richness, and evenness, which are the components that make up diversity, we have tertiary plots that show how all of those things changed in those different phases too. We wanted something that incorporated multiple metrics that we could then tease apart and look at what's driving changes in diversity. So in some parts of the river, for some taxa, it's richness. So the number of species has actually increased. In some, it's just sheer abundance. The same species are there, but they're more abundant than they used to be. So that's how diversity kind of is a multi-pronged metric that you can distill down into one measure, but you can also blow it up and look at it in more detail.
Just a quick comment on that. Shannon diversity of the other three, Simpsons and all those other ones, Shannon is the most relevant. It is the best. So that's what theory suggests also. That's just a comment. But because the student might have to leave, I want to ask if any students want to ask questions. Any questions from the students?
There was no data before, well, there is data, like I said, it's more scattered, more observational data rather than replicatable data. And also that was when we started to see the more broad scale anthropogenic factors starting to affect. Things were largely unchanged in Illinois, except for some small human settlements. Even Chicago wasn't what it is today in 1880. That was when we really started to see things start to change. So that was when we chose to start our timeline there. Good question though.
Back over there.
You mentioned something about Shannon in this, I mean, the diversity in this. So I want you to explain. Can direct my question to you.
Me? I'm not the speaker, but okay. Sure.
We can ask the speaker.
Shannon.
Shannon.
Like the Shannon is the case. So I want to know, is it possible to compare or to match all these indices together, Shannon, evenness, and the Simpsons?
So evenness is a separate thing, right? Shannon diversity and Simpson diversity would be the comparable things. But really, even so, Shannon diversity comes from this guy Shannon, who was a professor at University of Illinois, like 70 years ago. But anyway, Shannon diversity is not really a diversity metric anyway. It should properly be called something like Shannon entropy, right? But theory suggests that the diversity index, which would be something like the Hill number or something like that, the closest would be Shannon diversity. It has the best theoretical properties. Simpson's diversity does not relate to diversity, real actual diversity. So Shannon entropy does. So if I would have, done that, I might have done Hill number instead of Shannon entropy, right? But Shannon diversity is what a lot of people use anyway, even though it's not really technically a diversity metric.
I have one more question that I think applies. Sorry, I was watching
Sure.
on Zoom.
Yeah.
Out here. But are you involved in the Big River internships or summer positions?
We do hire, oops, minimize that. We do hire a lot of seasonal technicians at our field station, up to 20.
A lot.
Yeah, so we'll hire 3-monthers who have to go back to school in August. We'll hire 6 to 12-monthers that can work with us throughout the entire season. We've got 8 or 9 technicians at our field station right now who have overwintered with us, who were hired last year. So right now they're sectioning otoliths and processing zooplankton, people like Rachel and Abby who are picking macroinverts and doing things like that. So I'm not involved in that hiring,
Okay.
but I know people who are.
My other question, if you have any insight, so that's cool because it's lab work. One of my students was wondering if they needed to know how to swim to work with you all.
That's actually the first interview question that you will get asked if you apply for one of our jobs. Can you swim?
Yeah.
And
Is that a fail or pass?
it's not a pass fail. We all wear life jackets anyway.
Yes,
I'm-
I can't
I was
swim.
gonna say,
I said no.
I wasn't-
I would interview.
I wasn't going to out her, but Melissa can't swim. We all wear life jackets at all times on the boats. There's always more than one person on the boat for safety reasons. So if you can't swim, it's not a hindrance.
Okay.
I'm a triathlete, so I can help you learn how to swim. So,
Swim really good and far.
well,
A pass.
Yeah. or at least stay alive long enough.
Thank you.
Any other questions?
Students or otherwise, I can answer questions all day long. As long as my voice and my water hold.
I'll ask a question. So you showed how the community had changed from that orange dot to those other triangles and the diamond. So I noticed that the change is essentially going from more of a benthic system to more omnivores and carnivores in the recent times. But then again, towards the end, you showed that the food chain length had decreased and because of the heterogeneity decreasing. So I'm just wondering how you can have more carnivores but still the food chain length decreasing. So I feel like missing something here. Is it because, I don't know, maybe the total numbers of individuals have changed? Because that's not something you showed, right? Or maybe the biomass? I don't know what is happening.
We believe it's due to the benthification of the food web, the simplification of the food web. There are fewer trophic levels leading up to those top predators now. So there's not as much nitrogen accumulation. That signature has been depressed. So again, we believe that's due to the silver carp and bighead carp. Silver carp and bighead carp are planktivores. They suck down a ton of plankton. They have reduced plankton abundances in the Illinois River by over 90%. They're incredibly inefficient digesters. So they eat a lot, they poop a lot, and that poop is very nutrient rich. So they're affecting nutrient dynamics in the river. So they're taking all of this plankton, this pelagic mid-river water column food, crapping it out on the bottom of the river. And that's where all the food is now. So we've got organisms that are better adapted to feeding on the bottom. So things like channel catfish, they can just get their nutrients off the bottom. They don't have to wait for a shad to eat plankton and then die and then eat that shad. So the predators have not changed, but their trophic position has changed.
All right.
If you have more questions, maybe we can come to dinner, lunch with us. or-
Otherwise, my email's here.
We take the teachers and cookies and do something with it. Sodas. If you have a piece of pizza on the way out, please do some, otherwise I'm encouraging starving students